Yes, welcome back. We are still in animal cells and how they behave in a hypertonic solution. So, using the knowledge of osmosis, the movement of water. See, when we talk of osmosis, it is the movement of water from a hypertonic, a solution of low concentration, to hypertonic, a solution of high concentration. So this, the cell is more concentrated. The cell cytoplasm is more concentrated than the distilled water. So we can say the cell cytoplasm is hypertonic. The cell cytoplasm is hypertonic. Hypertonic to the distilled water. So the distilled water is hypotonic. The distilled water is of low concentration than the cell cytoplasm. So the distilled water is hypotonic to the cell cytoplasm. The cell cytoplasm is hypertonic to the distilled water. And therefore, if two solutions of different concentrations are separated by a cell membrane, then the one which is hypertonic will develop a suction pressure which will suck in water from the one which is of low concentration. So water will move by osmosis from hypotonic solution into the hypertonic solution. So water molecules will move by osmosis from a region of low concentration, which is the distilled water, into a region of higher concentration, which is the cytoplasm, through the semi-permeable membrane. So osmosis will occur. So if an animal cell, e.g. a red blood cell, is placed in a distilled water, which is hypotonic solution, water flows in by osmosis. Now, if water moves into the cell, what will happen to an animal cell? Remember, an animal cell does not have a cell wall, the rigid cell wall. An animal cell is only having the cell membrane, which is very weak. So the cell will start to bulge, to swell. Let's use the common language, to swell, to bulge. The biological language is bulge. The cell will bulge, and eventually, and lastly, finally, it will burst. The cell membrane will burst. Why? Because the cell membrane is weak. So that bursting of the red blood cells, when placed in a hypotonic solution, is what we call hemolysis. So hemolysis is the bursting of red blood cells when placed in a hypotonic solution. Why do red blood cells burst? They will burst because they do not have cell wall. They only have the weak cell membrane. So as more water is moving in, the cell membrane will bulge and finally it will burst. So that is what happens when the cell is placed in a hypertonic solution. Now, the third case. The third case is when we place an animal cell in hypertonic solution. So when an animal cell is placed in a hypertonic solution, what happens? What happens is here. So we are going to change we are going to change. And in changing, I will use, let us say that the cell cytoplasm is now 30%, for example, concentration, and this solution X is 50%. So what will happen in this case? So in this case, if we have the cell, the 30%, the cytoplasm is now low concentration, lower concentration. So if it is of lower concentration, then we say it is hypotonic. The solution X is of a higher concentration, then we say it is hypertonic. So the solution X is hypertonic to the cell. The cytoplasm is hypotonic to the solution. So if a similar red blood cell is placed in a hypertonic solution, then water will be drawn. Because now water will move from hypotonic to hypertonic through the cell membrane. So from hypo to hyper, water will get into the cell through the cell membrane. Now, the cell here let me repeat that. So if we have, let us say, if the cell, we are considering the solution to be hyper. So this is hyper. And the cell cytoplasm to be hypo. So what happens is that water will move from the cell. Let me correct that. Water will move from the cell. Why? The solution is hypertonic, but the cell is hypotonic. So water will move by osmosis from hypotonic, which is the cell, to the hypertonic, which is the solution X, through the semi-permeable membrane by osmosis. So this cell, animal cell, for example, red blood cell, will lose water. Water will be drawn out of the cell by osmosis. Now, when water is drawn from the cell will not maintain its shape. It will lose its shape and it will become, look that way, because now water is moving out of the cell. So when water moves out of the cell will shrink, and then by the process called crenation. So when a cell is placed in a hypertonic solution, the hypertonic solution will withdraw water from the cell by osmosis across the cell membrane. The cell will shrink, and then by the process called crenation. Now, so normally you find that to prevent crenation, in the body of living organisms, normally the body fluids that surround our cells, inside your body now, the body fluid is normally isotonic to the cells, so that there is no change. So this cell here, which has lost water and has changed its shape, is now described as crenated cell. So referred as crenated cell. So if you look at the picture that we have here, this one is a red blood cell placed in hypotonic solution. Water moves into the cell, it swells or it bulges and bursts. In the second case, So the bursting of red blood cells, we say, is called hemolysis. Then the second case is crenation process. How does crenation occur? The cell is placed, when the cell is placed in a hypertonic solution, water moves out of the cell shrink and become crenated. So that is a crenated red blood cell. So the opposite of hemolysis is crenation. Next time we look at how the plant cells behave in isotonic, hypertonic, and hypotonic solutions. Thank you for listening. Let us meet next time.